Let's go through these questions. And I'm in our normal classroom, so if I walk out of frame, it's just to change the next slide. Okay, so the first question that I got on this exam review was actually about stuff that we learned in the last section. But that's okay, the last unit, because this is an important topic, so I thought it was worth still addressing. So a hypothesis is going to be a specific and testable potential explanation for a specific question or observation. Okay. So this is like small scale. Okay, so you notice that ponderosa pine trees have their lowest branches are pretty high up on their on the trunk, whereas other species of trees that are of similar size have branches way closer to the ground. You can come up with a possible explanation that the ponderosas drop their low branches when they get to a certain size, so that to prevent fires from entering their canopy. So you can come up with an experiment to test your hypothesis that this is a fire adaptation, conduct your experiment, gather data to see if the data is going to support your hypothesis or not. Okay. A theory, on the other hand, this is broad. This is well established. This is tested with significant amounts of evidence. This is a framework for an observed phenomenon. That's how I'm going to spell phenomenon today. In order to have a theory, we need to have hundreds or thousands or more hypotheses. So here's our broad theory with a bunch of smaller hypotheses with the data that support the hypotheses. And all of these smaller hypotheses work together to provide evidence for this overarching theory for whatever the phenomenon is that we're trying to understand. Right, so our cell theory. Okay, we have an explanation for how new cells arise that's supported through evidence, through observation, through data. Everyday usage of theory means kind of in normal language, right? A theory, oh, I have a theory as to why the dishwasher is not working. No, bro, you have a hypothesis of why the dishwasher is not working. Okay, so everyday language, we kind of use these terms hypothesis and theory interchangeably, but in the scientific context, theory, we can kind of like, as close as we are going to allow ourselves, this is going to be fact, okay? But with the caveat that given new evidence, we might have to adjust our scientific theory based on new evidence. We're always going to allow ourselves to be wrong, but then because we always want to be building on the best evidence. Okay, all right, so let's move on to stuff that we're learning about now then. All right, so somebody wanted to know the main difference between prokaryotic cells, eukaryotic cells, and why prokaryotes and eukaryotes are important to biology. So prokaryotic cells are the more ancient form of life. This was what we expect. We think that life, like the first forms of life were probably prokaryotic cells or similar in structure and function to prokaryotic cells. And then later, eukaryotic cells evolved from prokaryotic cells. So these came later. And then this is kind of eukaryotic cells have a more complicated cell structure. Okay. So prokaryotes, these are the kinds of organisms we know as bacteria, and then archaea, which are kind of transitional. They have some characteristics of bacteria, but some characteristics of eukaryotes. And we won't spend too much time worrying about those details. When you guys take microbiology, you'll learn a lot more about the differences between bacteria and archaea. All kinds of prokaryotes are always single cell. Eukaryotes can be either single or multi-cell. So the only kinds of multicellular life are eukaryotic cells. You can't be a prokaryote and be multicellular, but you can be a eukaryote and be single cells. Okay. All right. So main difference: eukaryotes, which I'm just going to do the EUK. These have a membrane-bound nucleus. Okay. So that means that their DNA is stored in an organelle that has not one, but two phospholipid bilayers that's protecting the DNA, controlling what is entering the nucleus, what's exiting the nucleus, and all that stuff. So later on in the semester, we'll talk a lot more about the nucleus when we get to the DNA chapters. Eukaryotes also have membrane-bound organelles. And if you've already started working through the chapter 4 material, we're going to see that in the endomembrane system that has all of the organelle components that are used to transport stuff. The other important membrane-bound organelles that you're definitely going to want to know are the mitochondria, found in all eukaryotic cells, and then chloroplasts are found in plants and some types of algae. Or, well, I should say all types of algae, but some types of protists. So I will just write algae on here. There's other kinds of algae fit in that group of protists. That's kind of like the junk drawer of life. Like, I don't know. We don't know what to do with this. Throw it in the junk drawer, close it. Nobody ask questions. Okay, but there's other things like slime molds and amoeba and the other guy, his name is leaving my mind right now. I'll remember it when it doesn't matter. But those guys are also protists. Okay, so those are really the big differences. And if you guys want, we can go through the endomembrane system organelles. That includes the nuclear envelope, the rough and the smooth endoplasmic reticulum, the Golgi apparatus, we've got the vesicles and vacuoles. We got lysosomes in animal cells, lysosomes, and some others. Oh, and then of course, and always don't forget the actual cell's plasma membrane itself, which I should say a more accurate way to say that would be the cell membrane. Okay. Because of course, all of these organelles originally came from that cellular membrane. If we remember the folding in of the plasma membrane during the evolution from prokaryote to eukaryote. Okay, all right. Oh, I did not leave myself enough room to talk about the importance to biology, but I'll try. You know what? We can get rid of plants and algae. Boop, boop, boop. Okay. All right, so the importance to biology. These are, again, all forms of life are either a prokaryotic cell or a eukaryotic cell. Prokaryotes are the vast majority of the cell types. So like if we counted up all the cells, there would be more prokaryotes than eukaryotes by many times. Like I don't want to put a number on it because I'm not more accurate. So prokaryotes, again, the first forms of life. These are highly diverse. So there's lots of different kinds. So again, thinking about bacteria and archaea, these are very different ways of being a living thing. And then under each of those, there's a whole bunch of diversity. So we have lots of different species, but we also do have a diversity of functions. So what they're doing in the ecosystem varies a lot. And prokaryotes, always single cellular. And they're always microscopic. So you can only see them even with you need a very powerful microscope. The ones that we have at CCC are the ones that you might use if you're typically an NAU student. The ones that you'll use in your labs, you can see the prokaryotes, but you do need to use special techniques. So you'll learn that when you take that microbiology class. Okay, eukaryotic cells. These are again, the more complicated, more evolutionary advanced. Let me go back. So prokaryotes, we see evidence of those 3.5 billion years ago. Eukaryotes are about a billion years later. So these are more complicated. Again, can be multicellular. These include organisms like animals, plants, fungi, and protists. Again, defined by that, the membrane-bound nucleus. Okay, so question. So the DNA of a eukaryote is stored in the nucleus. Where in the prokaryotic cell is the DNA stored? I'll use red. And that's here in this nucleoid region. Okay, so oid means the form of or similar to. So the nucleoid region is like a nucleus in the fact that that's where the DNA is. It's not a nucleus because it is not membrane bound. Okay. So if you're going to remember one thing, eukaryotes have membrane-bound nucleus. Prokaryotes, their DNA is in the nucleoid region. Okay, so I know those words sound very similar, but sorry, you're just going to have to learn some vocabulary. Okay. All right, so this is where I got most of the questions. I had lots of people that wanted to go over this tonicity again. Okay, so and this is like the isotonic, hypotonic, hypertonic. Okay, and admittedly, this is confusing because there's a couple things that we have to keep track of in our minds. And this is why I really want you guys to have a scratch piece of paper with you when you're taking quizzes and exams, is so that you can scribble down and draw yourself a picture and logic this stuff out. Even if you can't, like, right, because it's not about memorization, it's about understanding the concepts. And so sometimes helping, like drawing a quick little picture, can help you logic out the answer, even if it's not right there on the top of your brain. You can get yourself there. Okay, so let's just remember that tonicity is the measure of the solution outside of a cell to change the volume of a cell because of this thing called osmosis. Okay, and I've gone back and forth in my career about whether I like this definition of osmosis or not. The reason that I don't like it is it's confusing. So let's read this for a second. Osmosis is the movement of free water molecules through a semi-permeable membrane according to the water's concentration gradient across a membrane, which is inversely proportional to the solute's concentration gradient. So hopefully everybody now understands osmosis, and we're so good and ready to move on. Okay, no, we're not ready. So let's break this definition down. We'll kind of look at those components because this is going to help us understand tonicity better. Okay, so osmosis. This is the movement of free water molecules. So when we're talking about osmosis, we're only talking about water molecules. Okay, now what do we mean by free water molecules? Well, if we have a ion here, right, because of the polarity, oops, I did that backwards. Because of the polarity of water, this partial negative charge of our oxygen is going to be hanging out with that. These water molecules that I'm drawing here are not free, they're trapped. Okay, they're hanging out with this sodium ion. They're not free, they're not hanging out all by themselves. But here's a free water molecule. Okay, it's just kind of not covalently or hydrogen bonded to anything else. They're just floating around doing their own jam. Okay, so that's a free water molecule. Okay, and it's important that we remember that there has to be a membrane involved in this situation. Okay, so the picture in your textbook was kind of like this little tube, and then they had like a dotted line to show that semi-permeable membrane. And we've got a bunch of, my red is going to be the solutes, so this is the stuff that's dissolved in the water, and then my black dots here are going to be the water molecules. Okay. Okay. So let's just remember the point of this slide then is just that there has to be a membrane involved. If I erase this, then it's going to be just simple diffusion where the solutes are going to equal out their concentration. But if we have a semi-permeable membrane in there that's preventing the solutes from being able to pass through, that's when we can start talking about osmosis. Okay. Okay. So the other half of this definition, okay, Now I got to draw my picture again. We said that water was black and the solutes are going to be red. Or something like that. Okay. So the water is going to move through the semi-permeable membrane according to the concentration gradient of the water itself. Okay. So on my right side, I have a high concentration of water. Okay, remember these square brackets mean concentration. Okay. On my left side, there's a low concentration of water. There's fewer black dots, right? So the net overall movement of our water molecules is going to go from the high concentration to the low concentration of the water molecules. Here's the confusing part where people get mixed up because this is frickin' backwards land. The concentration of the water is inversely proportional to the solutes concentration. Okay, so here's my little teeter-totter stick. Can you guys see that okay? So if I have a high concentration of water, let me do it the other way around. Okay, if I have a high concentration of water, that means that I have a low concentration of solutes. So on this side, if my water concentration is high, my solute concentration is low. On this side, though, I have a low concentration of water, which means that let me see, I said water is this side. So my water concentration is low. That means my solute concentration is high. Okay, so being inversely proportional is just fancy science talk for when one goes down, the other goes up. Okay, just like a teeter-totter. Okay, so hopefully we understand osmosis a little bit more better. Oh, let me say an additional thing. Okay, because when we are talking about tonicity, the words isotonic, hypertonic, and hypotonic are referring to the concentration of the solutes. So the net overall movement of the water molecules is going to go from the low concentration of the high solutes to the high concentration of the solutes, which is like backwards from what we're usually thinking of because we have to keep this inversely proportional idea in our brains. Okay. If I have a high concentration of water, I have a low concentration of solutes. My water molecules are going to want to move from high concentration of water, which is the same as low concentration of solutes, to an area where water concentration is low and solute concentration is high. Okay. I know it's a little mind melty, but this is why drawing a picture out is going to be able to help you. Okay. All right. So let's stop talking about osmosis and start talking about tonicity. Okay. We like isotonic because this is the easy one. Okay. Iso, we remember this means same. Okay. So here's our cell. This is where I need more colors. I don't have them. We'll have to deal with it. Okay, so here's our cell. Inside, the solute concentration is the same on the inside of the cell and the outside of the cell. Okay. The concentration of the solute inside the cell equals the concentration of solutes on the outside of the cell. Same-o, same-o. Okay. That means that water molecules are going to be moving into the cell at the same rate they're moving out of the cell. Therefore, the cell volume stays the same. Okay. That's the simple one. Then we have to do some mind meltiness. Okay. In a hypotonic solution, we're going to have a lower concentration outside of the cell than inside of the cell. Okay, so our solutes are red. Here's our cute little cell again. The concentration of solutes outside the cell is going to be lower outside the cell than inside the cell. 6, 7, 8, 9, 10. Okay, so that means that we're going to have let's see, let's make that 5 water molecules. Okay. 1, 2, 3. Okay, so in this case, our solute concentration is higher inside the cell, and our solute concentration outside the cell is lower, which means that the water concentration inside the cell is lower, and the water concentration outside the cell is higher. So which way is the water molecules going to go? The water molecules are going to move from an area of high concentration of water molecules to a low one. We're going to see a net movement of water molecules into the cell to get the concentration of water equal. Okay, hypertonic solutions. Okay, it's the opposite. Okay, the word hyper means above. So we're going to keep this concentration of solutes inside the cell at the constant. So solutes concentration. Well, let me do it this way. The solute concentration outside the cell is going to be higher, and then the solute concentration inside the cell is lower. So we're using this as our frame of reference for the terms hyper and hypo. Okay. Okay. So let's draw our solutes. 1, 2, 3, 4, 5. 4, 5, 6, 7, 8, 9, 10. Okay. If we have a lower solute concentration inside the cell, that means our concentration of water is going to be higher. 6, 7, 9, 10. And then outside the cell, we'll have a lower concentration of water. In this case, our water molecules are going to want to equalize the concentration inside and outside the cell. So we're going to see a net movement of water molecules out of the cell to get that water concentration equal. Okay. So in a hypertonic solution, an animal cell is going to shrivel up and die. A plant cell in a hypertonic solution is going to have their cell membrane, or sorry, their cell wall is going to stay nice and rigid, but their plasma membrane is going to shrink in volume and pull away from the cell wall. Okay, let me go back one slide. Okay, if we have animal cells like these red blood cells in a hypotonic solution, where water is entering the cell to equalize the water concentration. That because animal cells do not have a cell wall, their volume is going to increase and increase and increase as they take on more water until they have a cell explosion. Okay, the fancy word, science word for that is lysed. Okay. Okay, and that's because, again, animal cells lack that, any type of rigidity. Okay, in a plant cell, this plant cells are going to be taking on more and more water, but because we have that cell wall, all that extra water is going to be stored in the large central vacuole. And that's going to push the plasma membrane up against that rigid cell wall and actually give it more structural support from that trigger pressure pushing against that rigid cell wall. All right, let's move along then. Okay, so I have one student ask about bulk transport, so we'll do that real quick. And then we'll talk about those carrier proteins, which a lot of people have questions about. All right, so bulk transport, there's four kinds. Well, there's two major kinds, which is exocytosis and endocytosis. And then under that umbrella of endocytosis, we have three different flavors. Okay, so endo is in, we're going to move stuff into the cell. Exo means out, so we're going to move stuff out of the cell. Phagocytosis, we can remember that phago means eat, pino means to drink, and then we'll talk about these receptor mediated ones too. Okay, so that's just kind of an overview. We'll go through each of these flavors. All right, so phagocytosis. Okay, remember this is cell eating. Okay, and I've left this same picture up that was in the lecture slides and is in your textbook, but I'm going to draw it out so that we can kind of see the changes. Okay, so here's our cell. Here's a food particle. Okay, whether this is a bacteria cell or just a large organic molecule or whatever the cell is after. Okay, so the cell might move closer, but what the cell is going to start to do is just to pinch in their plasma membrane. Around that particle that they're going to bring in. Okay. This is my attempt at a time series. Okay. And eventually, the plasma membrane is going to pinch off to form a vesicle, which is a word I always spell wrong every time. So, is that spelled right? I don't know. Okay. Now, this food particle is within the major cell, right? The main cell. Let me erase that because that kind of. So now we can have another specialized vesicle called a lysosome. So I'll just have an L come here, right? And then these vesicles can fuse together, and then all the enzymes that are in the lysosome then can go and digest and break that food particle down, whether it's another bacterium or another cell or just a chunk of organic material. Okay, so kind of now we know like what's happening, and this major thing of like creating a vesicle is pretty much how we're going to do all of the phago, pino, receptor mediated, and exocytosis. Okay, so pinocytosis is where we have a bunch of small solutes kind of in the extracellular membrane. So this is outside the cell. So we have all this stuff in there. So the cell is going to do the same thing. Right, here's all of our solutes that are floating around. The cell kind of wants to do like a little taste test to see what's out there. So they're going to pinch in a little, a little vesicle to bring in those solutes. They're going to pinch that off. Let me draw that more better. Okay. And then they'll eventually pinch that off to create the vesicle. Then they can kind of see, okay, what of all the different kinds of solutes that are out there, which ones do we want to bring in? Which ones do we want to keep out? Okay, so it's a taste test. I think in the lecture videos, did I use the analogy of getting a flight of beers where you get like a bunch of little tiny beers and you taste them all before you commit to a big beer? All right. Okay, so after we know what solutes are there, if we want specific solutes, then the cell is going to transition to using this receptor-mediated endocytosis. Okay, so in our cell, remember, we have all these proteins that are sticking out of here, and each of these proteins has a specific shape that is going to fit whatever our target molecule is. So things that are triangle in shape can't fit in here. So they're going to try and come in and they're going to get boinged off. Okay, so oops, wrong color. So we're going to do that same thing again, where we take that plasma membrane now with all these proteins that are embedded in it. We're going to pinch that in to the cell. So I'm going to redraw all this crap. Okay, pinch that off. All that stuff is now on the inside of this vesicle. And we've only picked up the circle solutes and left all the triangle solutes outside of the cell because we don't want that stuff right now. Okay. So this is how we're going to get, we're going to bring in specific stuff. Into the cell. So only the things that we want. So your neurons are a great example of this, where we have specific proteins that are going to bind with those neurotransmitters and nothing else. And then we can have these vesicles uptake those neurotransmitters so that they can be recycled. Then exocytosis is the same process, we're just doing it backward. Okay, so maybe after we phagocytosis a bacterial cell into our, the host cell, we break down that bacteria, that food particle, and now we've created a bunch of waste products that we don't want in our cell. We got to get rid of it. So we're going to have inside the cell, we're going to have a vesicle with all the waste products. This vesicle is going to move and fuse with the plasma membrane. And that's going to allow the export of all that stuff the cell doesn't want in its body. Okay. Because the plasma membrane of the cell and all these vesicles are all made out of phospholipid bilayers, we can do that, pinch them off, reabsorb, move the things around in the cell so that we can make sure our cells stay nice and healthy. Let's talk about these carrier proteins then. Okay, a few people had questions about our different kinds of carrier proteins. Let me move that mouse so that I'm not distracted by it. Okay. So we talked about 3 different kinds of carrier proteins. And remember, these can either be active transport where we're using ATP, or they can be found in passive transport as well, where we're just using the concentration gradient. Okay. We tend to see them more often with active transport, though. All right, so let's talk about our 3 different flavors. So our unitransport protein, so just like a unicycle has 1 wheel, a unitransporter is going to transport 1 solute. Okay, so we're just taking we're taking our solute from the exterior of the cell. We're going to have it. Okay, and again, this is time 1, and here's time 2. So we're going to see this change in the 3-dimensional shape of the protein. So this is my crappy drawing of that, where now we can deposit that solute into the cell. So I guess I should have drawn, here's my phospholipid bilayer. Okay, so we're just moving 1 solute at a time. That one's easy. Okay, our symporter, just like a symphony, we're all playing, we have all these different instruments that are playing the same piece of music. So sim means same. So now we're going to take 2 solutes and we're going to move them in the same direction. I know how to spell words. I don't. Okay, so this can either be both solutes coming into the cell or both solutes going out of the cell. Okay, so let's have my protein wants to be like this, I think. Okay, pretend this makes sense. Okay, here's my plasma membrane. In order for this protein to become activated, we have to have solute 1, let's pretend that's a triangle, and solute 2, pretend that's a circle. Okay, and then once both solutes are docked into that, then we have the change in the 3-dimensional shape depositing these both solutes either into the cell or out of the cell, depending on where we're going. Okay, so sim means same. We're taking 2 solutes and we're moving them in the same direction. Anti, that means against. So here we're going to take 2 solutes and we're going to move them in opposite directions. Okay, so let's try to do a triangle here on this side of that protein. This side of the protein is going to have the circle. Here's our plasma membrane. Okay, so our triangle is going to come and dock in there. On the inside, our circle is going to come in here. The triangle is going to go this direction. The circle is going to go in that direction. So in the case of our antiporter, we're going to take 2 solutes and we're going to move them in opposite directions. Okay. And we've seen a few examples of those with the cell communication stuff. So be looking for these examples as you're going through the cell communication stuff.